Mark is going to look at and talks about the whole area of research, but he's also he's principal investigator within the Cork Cancer Research Centre, where he heads a research team investigating various anti-cancer gene therapies. His interests include developing new, safer ways to treat cancer using microbes that specifically grow within tumours. His research in this area has attracted significant media attention internationally and recently as well, I believe. He's a frequent guest speaker at international conferences, and he's joined forces with a large network of like-minded laboratories around the world. And as a result, this collaborative research is achieving rapidly towards patient treatment. And this is something that I suppose we always want to see. What's the latest thing happening in the whole area of research? So, without further ado, over to you.
Thank you very much. You can give a longer introduction if you want, because my laptop has just decided to restart and take its time, so I could just tell a few jokes or something. It won't be as funny as Seamus, but So anyway, it's an absolute pleasure for me to be here. I work in the university here in Cork, University College Cork, and the Cork Cancer Research Centre is based on campus. So what we are is that we're an entirely research-focused centre. We exist purely through research, in that we don't do any academic teaching, so we're full-time at the research in the hope of focusing ourselves entirely on finding new treatments for cancer. The type of new treatments we have, again, I suppose Seamus was glad to say used an analogy of cancer treatment developing using cars as an analogy. So he's absolutely right in terms of the difference and the improvement in chemotherapies in 20 years is absolutely correct in terms of a Volkswagen Golf in the 70s compared to a modern one. At the same time, there's always room for dramatic improvements as well. So for example, I've heard the phrase before that if Henry Ford, before he developed the affordable car, asked people what they wanted, they would have said a faster horse, which would have been an improvement and incremental, but at the same time I do think there is a place for more dramatic technology and an improvement in what we do. So that's kind of the space that we're working on. In that we're trying brand new, absolutely novel therapies. So you'll appreciate I'm waffling a bit here just to try and get my slides up. The Cork Cancer Research Centre, we started at the University Hospital more than 10 years ago now, and the late Professor Jerry Sullivan was our founder, and he realised that the typical Irish cancer research, which is largely based in universities working on their own and not really communicating with the medical profession, is wrong, and that what we need to do is to have a centre to combine people like Seamus, the surgeons, the patients, and the research scientists like ourselves. So what we have is an attempt to get this is a conduit between the various partners, and exactly as Seamus said, everybody needs to be a partner in this. No individual can do this in isolation. So we started in the Mercy, we then expanded, and we have the top floor of this building is the Biological Sciences Institute in University College Cork, and we still work with people in the Mercy and in the CUH as well. And we kind of act as a hub for research. So this is our lab on the top floor. And I suppose one of my main jobs that I see here today is just to kind of explain how cancer research works. Because people who aren't involved in research won't really have an understanding for it. I do think they should know, because we are so dependent on the fundraising that people do for us, and we like to make sure that it's first of all being used appropriately, but also how it is being used. So this lab, anybody's more than welcome to come visit anytime they wish. Could be any research lab in the world. If you walk into the States or in England or anywhere, they all look exactly the same. And the only reason I mention that is just because people might think, oh, we'll never be as good as the cancer researchers in the States and elsewhere. They've got so much money and they're so much more intelligent than us and better looking, etc. But in actual fact, we do pretty good. We have exactly the same technology and skills and education as to do anywhere. So it is worthwhile pursuing cancer research here in Cork, in addition to all the research that goes on abroad. And we partner up with people abroad. We absolutely realise we can't do it on our own, but we have a niche, and people realise that and they capitalise on that, and therefore we can offer something which other people around the world can utilise with their own. In terms of our sources of income, I don't get to do much science anymore. I just spend all my time begging for money, mainly from national government grants or charity agencies, but also international increasingly as well. So we try and get as much money as we can from the EU and the various cancer and general science funding agencies in Ireland, but also we're extremely reliant on charity funding. So just to explain, the Cork Cancer Research Centre's charity fundraising wing is called Breakthrough Cancer Research. So Breakthrough Cancer Research is a kind of a national entity, and it does all the fundraising, and they have a wonderful machine for attractive philanthropy and fundraising, and that goes to support the research in the Cork Cancer Research Centre. So broadly speaking, just briefly to say how does science research work. So this is true for any type of science or any technology development really. So some scientist or doctor has an idea, and the first thing they do is to go and get some money to try and do that. That money then feeds into the laboratory where research takes place by the guys in the lab. This goes on for some time in various projects and various people and for a length of time. Then eventually you get new technology. So somewhere in towards the end here, you also have, let's say in the cancer space, you've got clinical trials in the hospitals, and to actually make the medicine, you eventually have to have the big pharmaceutical companies involved in as well, because it's only they can actually afford to do the proper final clinical trials and genuinely make an actual medicine. So this is the cycle that we typically undergo. While doing this, there are three stages of technological advance for any technology. It always starts off with the naysayers will say, forget it, it's just impossible. But that's kind of said for everything, and if people listen to that, there will be no technological advance, and people are just not bothered doing research and nothing would happen. So eventually when people plough through it, and I've lived here for a long time until a few years back, next stage is like, look, it might be possible, but it's just too difficult, it's not really worth doing. So a lot of my work now is kind of at the later stage along here in generating convincing evidence that it is worth doing. It is medically feasible and worth doing, it's commercially feasible and worth doing, and raising money to go and do that and actually executing the science as well and developing it into an actual medicine. Again, I stress, we don't do basic research for the heck of it. We are very much applied research in terms of taking knowledge that's been generated over decades by various people around the world and trying to put it into patients. And then eventually you'll get the smart artists at the end who go, oh, I knew that was a good idea all along. But they're the very same people who said forget it, it's impossible. So it's always a struggle. It's a financial struggle, but also it's a kind of a mental struggle just to keep going. If you know you're onto something good, you keep going. That's kind of the daily life of the scientist. So in terms of development of new ovarian cancer treatments in Cork, again, I just want to say that look, this is happening under our noses and on our doorstep, and I'll just use my own group in Cork as an example, but we're pretty typical of what's going on in any research centre around the world, in Dublin, in the States, and in anywhere. So basically, as you probably know, how a tumour forms first day is that, as Seamus pointed out, something happens to one of our healthy cells. Our healthy cells are destined to die, and that the genes within our cells are the instructions for our cells, and the eventual instruction to all our cells is to die, because you're supposed to be replaced by a new cell. That doesn't happen in cancer, and you get a build up of these cells that just refuse to die. every single cancer treatment out there is based on trying to find a difference between our healthy cells and the cancer cells. Every single treatment, that's what it's all about. We've got plenty of toxic chemicals that will kill a cancer cell, but in doing so they'll kill us as well. So all our cancer treatments are directed at finding these subtle differences between the cancer cell and the healthy cell. So where we're working in this space is developing new advanced therapeutics, which are what I like to simplify down to is that we're trying to deliver a message to cancer that ultimately results in that cancer's death. So the analogy here I'm going to use is that the envelope is the messenger, and the piece of paper inside it is the message, and this varies. So for example, we have developed systems where we can deliver a message to a cancer cell, and the message inside the messenger is self-destruct. So we try and get these into cancer cells so that they just kill themselves. Alternatively, instead of delivering it directly to a cancer cell like we did a while ago, you could deliver to the immune system, and the message in this case, when we deliver it, is here's a cancer cell, kill it wherever you see it. And that is what immune therapy is, and this forms a large part of our work. The benefit being that, for example, if you have delivery to cancer cells, theoretically you should treat every single cancer cell, which means that your messenger has to get into every single cancer cell, which is difficult, but a very, very powerful approach that now people are absolutely realising and is now getting into patients and now works, is that if you can just teach the immune system to recognise the cancer cells, then they will go and do all the work, and the immune system is constantly surveying for the rest of our lives, and they will go and seek out and destroy any cancer cell anywhere in the body. So how we do this is that we work largely on the messengers. And one example of what we do in our lab is that we use bacteria as messengers. So the reason we do this is that people for a long time have known that bacteria are pretty good at getting into our body. So bugs, we all suffer infections normally, and bugs or bacteria or viruses, and they're pretty good at getting into our body. So we can hijack that. When it comes to bacteria, so typically if a bacterium gets into your body, the immune system recognises it and kills it pretty quickly. If it's a disease-causing bacterium, you might get sick for a while before the immune system kicks it out. We use non-disease causing bacteria. We actually use like probiotics, which are commonly found in yogurt and cheeses, and you can make these capsules and stuff. So we use variants of them. The reason we do that is that they lack the ability to cause any disease. So normally if you were to introduce them into the body, if you were to inject them in, the immune system would just kill them straight away, except in tumours. So tumours differ from any healthy part of the body in that they're the only place in the body that lacks the ability to kill these bacteria. So because of that, the bacteria love to go there, they can't die inside there, and they grow to huge levels inside there. They set up home, and they keep replicating inside there, and they keep growing. So just a video on that, hopefully, is that if you were to give an intravenous injection, for example, that's not it, of a bacterium into the body, it will circulate around the body but be killed in all the healthy parts of the body. So when we do this, we see that the bacteria don't survive for more than a couple of days, except in tumours. So it doesn't matter where within the body that the tumour is, as long as there is blood supply to it. So the bacteria go in, they set up home. So the key here is that your regular bacteria won't do anything. Eating yogurt won't cure cancer. We need to modify the bacteria. So Ireland has a history of agricultural research and dairy research, and Cork in particular has been a world leader in the bacteria involved in food making, and that. So as a result, by coincidence, it was never meant for cancer research, but Cork are kind of world leaders in technology to genetically engineer these types of bacteria, and we know how they work extremely well, better than anybody else. So in this context, that's our niche, and that's why people around the world are more than happy to work in conjunction with us. So we genetically modify these bacteria to make anti-cancer agents. So as you can see there, when the bacteria get into the tumour, they pump out these anti-cancer agents just within the tumour because they can't survive anywhere else in the body. And that is in effect how we address, as I mentioned, every single cancer therapy is trying to find differences between the tumour and the healthy part. This is how we're doing it. So we specifically deliver our message just to the tumour, and in there it goes and instructs the tumour to die. We also use it for immune therapy, and in doing that it can, for example, locally flood the tumour with these agents that call the immune system in. And once immune cells are in the tumour, they recognise the tumour cells, and then they can get educated to go and recognise and kill that cancer more. In the context of ovarian cancer, we've done this strategy in a number of cancers. We absolutely realise we can't do everything, nor do we want to do everything. So we pick our best targets. And in recent years, we've realised that ovarian cancer is one of our best targets for the strategy. The nature of the tumours just really permits our bacteria to grow in them very, very well. And the other angle unique to ovarian cancer would be this unfortunate occurrence of ascites. So you get this build-up of fluid in the peritoneal cavity occasionally with ovarian cancer, and that fluid in the abdomen is a perfect home for our bacteria to grow, we've discovered. So Seamus pointed out earlier, you can give this direct injection into the abdomen to locally treat where the ovarian cancer is. We've realised now that our bacteria are particularly good at that, and that they don't leave that area. So if you do give a direct injection in there, our bacteria can't go anywhere else, but they will live in there very happily and pump out their anti-cancer effects. It also works very, very well for immune therapy, so we can educate the immune system to the presence of the ovarian cancer cells there as well. So yeah, just for finishing up, in terms of, again, just trying to impress upon people that we're every bit as advanced here in Cork as we are anywhere in the world, but one of our kind of leading technologies that we're kind of ahead of the game and internationally is imaging for research. So what we have here, there's no way you'll recognise it unless I try and explain it as best I can. So the brown area here is basically a tumour. So this little roundy lump here is a tumour, and what we've done is that we've made the cancer cells emit light, and that light is green and blue. And we've made, so this is real, and we've made the bacteria emit this orange light. So the reason we want to do this is that we want to see in reality, going beyond the test tube or any theoretical, where are the bacteria in relation to where are the tumour cells and what's going on over time. So you can see the blood vessels are around the tumour here as well. So this is absolutely direct evidence to us when we're doing our research. It informs us in real time. This isn't theoretical, this actually happens. Here are the bacteria, we know exactly where they are, we know how long they last, we know how they spread. We know that the tumour cells become less blue and green and give less light as we kill them over time. So we have technology here that a lot of people around the world don't yet have. And because of that, we have partnerships with, for example, the company that makes this technology. And as a result of that opens up our network and our opportunities for doing work like this. So, and again, this is just, again, impressing, not trying to blow my trumpet, I'm just saying that in Cork and in Ireland, we can do stuff that makes the newspapers and stuff every bit as well as the international labs. Just in the context of it's worthwhile us fundraising and raising money locally for it to be spent locally, and that it's not just a waste of time that sure America is just going to cure cancer and they will never do any work here. We are doing our part. It's a universal approach. It's kind of the international cancer fight is the way that everybody approaches it, and only through this international cancer fight can we inform everybody else of what we're doing. So like any work that Sharon's group does in her lab in Dublin informs our work and vice versa, and she's experts in how cancer cells grow. We take her knowledge and then we can deliver things into the tumours for her and work like that. And then just to finish up, again, blowing trumpets and how people around the world like us. This was a visit a couple of years ago from this man here was Dr. James Watson. He was one of the 2 people who got the Nobel Prize in the 1960s for discovering DNA, or how DNA works, and how he was willing to travel from the States to Cork just to see how we work and to interact with us as well. So it was a couple of years ago, and I suppose what struck me recently is that the importance and the urgency of us to proceed with our cancer research work, and that 2 of the people in this photo have since died of cancer, and it's not the old guy. So we really do need to, when we see patients, and when we see the need and the urgency, it really does impress upon everybody in the lab to go and work as hard as they can and as quickly as we can as well. So that's it. Thank you very much for your time.